So this is the last video on chapter 5, cell division. And in this chapter, or in this video, we are going to be talking about the second division of meiosis. Okay, so meiosis 2 is very similar to mitosis. We're still going to have the same PMAT, just like with mitosis, meiosis 1, meiosis 2 is going to have it. So in prophase 2, cells only have one chromosome from each homologous pair because they were split in meiosis 1. We're going to have the nuclear envelope is going to be disassembled, and each duplicated chromatid attaches to the spindle. Metaphase 2, they're going to line up in the middle. Anaphase 2, the sister chromatids are going to separate, so line up in the middle. Metaphase, anaphase, they pull apart. Telophase 2 is just like telophase and mitosis. The nuclear envelope reforms, and then cytokinesis is going to occur either with the cleavage furrow in animal cells or the cell plate with plant cells. Okay, so in meiosis 2, we're already at N because meiosis 1 went from 2N to N. So meiosis 2 is just going to go from N to N. Okay, so prophase 2, our nucleus envelope is going to disappear. We have our centrioles forming. Metaphase, they line up in the middle. Anaphase, they pull apart. Telophase, we start to get our nucleus together, and then finally cytokinesis, we are going to have 4 haploid daughter cells. So they have half the number of chromosomes as the very first cell does, and there's 4 of them. And notice that they are genetically different as well. So meiosis produces haploid cells from diploid cells. It's important because it helps with genetic variation, and it occurs in crossing over in prophase 1 and independent assortment during metaphase 1 and anaphase 1. Upon fertilization, combining of chromosomes from genetically different gametes help ensure offspring are not identical to parents. This genetic variability is the main advantage of sexual reproduction, and long-term genetic variation increases the survival of a species. So genetic variation helps a species to survive longer. If every single organism in a species looked the same, if something were to happen, you know, one gene or trait would not be beneficial, then the whole species could go down. So genetic variation is helpful for a species to survive for a long period of time. So now we're going to compare meiosis with mitosis. So DNA replication occurs only once in either meiosis or mitosis, and this occurs in the S phase of interphase. Meiosis requires 2 division, mitosis requires 1. Meiosis produces 4 daughter cells, and mitosis only produces 2. The 4 daughter cells from meiosis are haploid, and then the 2 from mitosis are diploid. Daughter cells from meiosis are genetically different, while those from mitosis are genetically identical. So when does this occur? Meiosis occurs only at certain times of the life cycle of sexually reproducing organisms. So remember, meiosis is going to create sperm and eggs. So for creating sperm, sperm gets created all the time. Males can create sperm starting at puberty all the way through their death pretty much. Whereas with females, meiosis is only going to occur during development of the fetus. Female baby is born with all the eggs that they're going to possibly have. So it occurs at different times for different sexes, but mitosis is continuously, it's always happening because you're going to have cells that are going to need to grow and repair. So comparison of meiosis 1 and mitosis. In meiosis 1, you have the homologous chromosomes and crossing over occur. but not in meiosis. Paired homologous chromosomes align at the middle in meiosis 1, where just in mitosis, you're going to have sister chromatids align. And then in meiosis 1, during anaphase 1, those homologous chromosomes separate, and that's when you have a reduction in the chromosome number. But in mitosis, the centromeres split and chromatids are moved to opposite poles, and you have the same number. Okay, so this is comparison of meiosis 1 and mitosis, what we just went over. Now with my meiosis 2 and mitosis, these are going to look very similar. So prophase, no pairing of chromosomes. Metaphase, haploid number of duplicated chromosomes at metaphase plate. Anaphase, sister chromatids separate both of these. Telophase, 4 haploid daughter cells, not identical to parent cells in meiosis 2, whereas mitosis, 2 daughter cells identical to the parent cell are going to be produced with that one. Now, let's talk about the human life cycle, because both mitosis and meiosis are needed in order for the human life cycle to take place. At fertilization, a haploid sperm and a haploid egg fuse. The result is a zygote that has a diploid number of chromosomes, because you're adding N plus N is going to give us 2N. So you're going to create a zygote, which then is going to turn into a fetus and a baby. The fetus divides by mitosis for growth and development. So after birth, mitosis then allows for continued growth and repair. So you need both of them. Now, meiosis that occurs in males, it occurs in the testes, and it is called spermatogenesis. It produces sperm. Like I said, it starts in males at puberty, goes on for the rest of their life. You could have men, 80, 90 creating children because they're constantly creating sperm. Meiosis in females occurs in the ovaries, and it's called oogenesis, and it produces eggs. And eggs are only produced in the uterus of the mother when the baby's in there. That's when that happens. Okay, so right here we have the mom, 2N, dad, 2N. They go through meiosis. Oogenesis creates an egg that's haploid. Spermatogenesis creates a sperm that's haploid. At fertilization, you get a 2N zygote, which becomes a baby. Okay, and then mitosis, they are going to grow into adult human beings. So that is the end of chapter 5.